Hello everyone, this is the first tutorial on large language models for DNA. I'm Raphael Mourad, I'm an associate professor at the University of Toulouse in France, and I decided to make videos for illustrating the new GitHub repository that I've made on training large language models for genomics, especially for DNA, but it can be applied to RNA, for also protein, peptides, etc. So this is the GitHub. Uh, it has been made for everyone that who can use it. So there is a short lecture here that you can look at, and I will make at some point a video regarding for the lecture. And then I prepared some data here, and you'll find five tutorials, five lab classes. So first one, pre-training an LLM for DNA sequences, that I will do it just right now. Um, the second one on fine-tuning an LLM for DNA sequence classification. So once you have pre-trained your LLM, then we'll see how to use it. First for fine-tuning. Secondly, for prediction of a mutation effect with zero-shot learning. Then synthetic DNA sequence generation, meaning that you're going to generate artificial sequences. And finally, optimization of DNA sequence. So here is a repository. It's free to use. And then I have, in this repository, you'll find links to Google Colab that I've made so that you can reproduce. I mean, you can do all of the experiments of the lab class for free without requiring any special computing uh, computer. So uh, here is the first. So I clicked here on this first link, pre-training an LM for DNA sequences. I clicked here, and then you'll come to this page. Okay. So it's written, this is a script to pre-train a large language model that I've done, that I've modified, which is a the mixtral model that has been developed by the French AI company named Mistral. And this LM is pre-trained so it's a causal language modeling, meaning that given a sentence, so for instance, DNA, you'll predict the next word, so the next piece of DNA. So we'll see how it works. And if you're interested also, by the way, you can look at my Hugging Face page where I deposited a lot of models that I pre-trained. So for instance, you can see that I developed a Mistral model for codons. It can be used actually for mRNA, especially for instance for mRNA generation or mRNA optimization for a vaccine. I developed also models for DNA sequences. I trained on the DNA human genome from the human genome. Also trained on different genomes like Aryopsis thaliana, so plant. I made special models, specialized model for link RNA, for virus, for non-coding RNA, for peptides here, for bacteriophages, which are viruses that infect bacteria. So it can be interesting if you want to develop new antibiotics for plasmid, for yeast, bacteria for the human genome. Also, I developed a model for proteins. So, this collab can be used to pre-train a very big model. Here, of course, due to limited time of computing due to a free Google Colab, we will not run the training of over a big data set and over a long time. We'll just stop the training at some point, just after starting the training. So here what we do, first of all, this Google Colab is attached to another GitHub, which is called Mistral DNA. Uh, so where you can find scripts, you can find data, you can find a lot of things. It was the initial, let's say that it was initially also a training. So we'll just download this one first because there is some data that we want inside. So let's download it. So I just clicked here, and with git the git clone command, it will download the Mistral DNA Git repository on the server of Google Colab. So let's do it. All right. Then we have to install. So in Google Colab, there are many Python modules that have been already installed that you don't need to install, but there are few that needs to be installed. So I have to install datasets and flash attention. So let's install them. It takes a little bit of time, so we'll wait a bit. Dataset is quite quick, but then flash attention takes a little bit more time. It says that there is some conflict, but that's not that important. I didn't try to solve this part. It's not a big deal. So installing Flash Annotation will require installing cuBLAS, cuDNN. So that's why it takes time. So let's wait. So let's just say a few things on the while it's installing. Let's say let's talk about a few things. So first of all, you have to look at the runtime if you want to. So you can run this Google Colab on the CPU or on the GPU given by Google. So what you have to do is normally by default I think it's a CPU here. So first of all, you can change the running time, which means the code, the language, the programming language. You can either pick Python 3 or R. So here, of course, it's a Python code, so you can select Python. And then here, this is the choice you have. You can either select a CPU, where the time of computing is almost unlimited, or you can select a T4 GPU, where the time of computing is quite limited. Also, or a GPU. Also, you have the choice of using a TPU. I didn't use that. So here it is. You can, if you want to run on GPU, use T4 GPU. And the code has been developed for being used on GPU, but you can run it on CPU. That's not that's not a big deal. So everything has been installed now. Let's check that you see the GPU, the T4 GPU. And indeed, you can see T4 GPU, and this is great from Google that they provide access to a real GPU. And you can see that you have 16 gigabytes of VRAM, which is not a bad amount. Here's the CUDA version that you have, 12.4. This is a driver version, okay? So this is quite important because if you don't have the right CUDA version given a Python module, you can have sometimes incompatibilities. So let's load the libraries. So we need classical library from OS, okay? But so the OS library from Python, but you need most importantly, you need the Torch library for deep learning, NumPy for manipulating handling data, and the transformers library for that was built on the top of Torch for training large language models. And also we're gonna use the library dataset for making a dataset for the network. So let's load that. Everything should work. All right, so it's loading all of the functions that you need. All right, let's check the NumPy version because sometimes there are problems. So I checked the versions here so that everything works. Transformers, okay. This is not the latest transformer version, but it works fine. Flash attention, here it works. Accelerate to compute on a GPU. Then here it's to constrain the memory. If you want to see that if you really downloaded the Mistral DNA repository, I'm just doing an ls to check that I have it. And then I will set the directory inside Mistral DNA. And then let's start. That becomes interesting here. So how do I load a model? So the model architecture. So inside data models, I have a folder that contains Mistral architecture that I've simplified. I'm gonna load this JSON file. I can make you have a look at this JSON file. So this is here, models, and here. So this is just the JSON file that you need, the JSON file that you need to configure your large language model, the Mistral model. So the most important file is this one. So it says that the architecture is Mistral for causal language modeling. It tells you the size of the neurons inside the network. So you can see that the original Mistral model has a bigger hidden size actually. I reduced it for being able to run the pre-training on the model on the Google Colab. So it's a very simplified architecture that you have here. I just change the numbers here and here and to make it able to be trained on the Google Colab. So the intermediate size also is 256, so it's pretty small. The number of session head is only 8. The number of expert is only 1. The expert per token and the number of local expert is 64. And the vocabulary size is not something like 50 or 60,000, what you can find for English or French or languages. It is for DNA, and 4096 corresponds to kmers kmers of size 6. So for instance, 4 to the power 6. So it means kmers of type 6, because you have 6 letters in a kmer, so ATGC. Kmer of type 6. So for instance, a kmer of type size 6 is that. Wow, this is a kmer of size 6. Okay. So we will model the DNA by looking for kmers of at least size 6. So this is a vocabulary size. So yeah, we'll see after the byte pair encoding. So so this is the configuration model, and then we will load the model using the configuration. So the model architecture, this is not a pre-trained model that we'll load. We'll just load the architecture. So without the weights of the model. For that, we'll use auto model for causal LM. And here, we'll not use flash attention. We'll just use simple attention. So here you can see that you have 8 layers. This is an LLM with 8 layers. You can see that originally you will have a dimension of 4096 words in your DNA sequence. And it will be converted to layers of only 256 neurons inside. And then we'll output, at the end, we'll output 4,096 words, because the idea is to take given a DNA sequence to predict the next word. So you have to be at the same dimension as the input data on the output. And then we'll use to make words, we will use byte pair encoding letter tokenizer. So we'll use the one that has been done in DNABird2, but we can make also our own. I didn't include the code to make your own tokenizer. So let's load the tokenizer. So here it is. I mean, we can have a look at this tokenizer if you want also, by the way. This is a tokenizer JSON file of the tokenizer, and we will see. Because it's already included, the tokenizer is also included inside the model files. So what's interesting here is the following. This is a tokenizer of the English, sorry. The tokenizer we have is different. So you can see, yeah, I will take an example. So for instance, you can see special characters, for instance, unknown, separator, paddling, mask. And if you take a small sequence like ATT, GT, GGGGT, CCCC, we'll tokenize it. So it will be tokenized as follow. This is what the network will see. Just a 1, 2061, 281, 485, 6, and 2. So it means that this is the code of each subword of the sentence. And I can change it if you want. Let's make something simpler. ATT. So ATT will have this code. 1 means the beginning, CLS means the beginning of the sentence. 2061 corresponds to ATT, actually. And 2 is the end of the sentence, a separator between sentences. So it's the end of the sentence. So here is the beginning of the sentence. Here is just ATT is encoded as 2061, and 2 is the end of the sentence. Let's now look at how many parameters there is in this model. So there is 105 million parameters. So you can see it's already a big model. We'll load a very small data. So what I've done is I pre-trained a Mistral model on the whole human genome. But here, so I've trained it, and then you can see it on my sorry, let me just adjust my window. You can see it here. So for instance, this is a Mistral DNA model that has been trained on the whole human genome, which is here it's a small model of 1 million parameters. Sorry. And here I didn't take the whole the whole data because it comprises too many sequences. I took a small amount of data, so it's just less than 1% of the DNA sequences from the human genome. So we'll load them. And this is here just the first 5 DNA sequences. So this one contains a lot of repeat elements. This one also, because this is beginning of the, so this looks like telomeres, I guess, because this is the beginning of the chromosome. Then I have a tokenize function that will tokenize all of these sequences so that the language model can understand the DNA sequence. So I'm just tokenizing all of the sequences. You can see that's quite fast because I have 99, almost 100,000 sequences. So the sequences are 200 base pair long, just to say. 200 base pair long, and we have one almost 100,000 sequences. So that's quite quick to tokenize all of this data. And here, what I'm gonna do is I'm gonna split the data into a train data set, a test data set. I mean, and also, yeah. So the train, yeah, I just named the train dataset and it's called the validation dataset. So I'm going to split the dataset in two pieces. Uh, let's look at the data that has been tokenized. So this is, for instance, the first sequence of 200 base pair here, and here these are the input IDs. Okay, so these are the token numbers, the token IDs. Okay. So let's look at the train set. So almost 8,000 sequences. And here, what I'm gonna do is make to save the training parameters inside the function training arguments. So I will save the results inside the folder result models. The evaluation strategy will be by epoch, meaning an epoch is a time that you've seen the data. So each time I see all of the data, I will evaluate the predictive ability of the model on the validation data. Save strategy each epoch. So after each epoch, I save the model. How many training epoch should I do? So I made 50 training epochs. So 50 times I will see the data. And this will allow me to optimize the loss, to minimize the loss basically, until it reaches a minimum, a local minimum. So this is the batch size. So I picked a batch size of 32, not too big because cannot be computed on the Google Colab. A learning rate of 5, 10 times minus 4, which is quite standard for pre-training. A weight decay of 1%, quite standard. What's important is here, load best model at the end. So during the gradient descent, you will optimize your model at each epoch. And then at some point, the model will start to, the loss will start to increase again. You want to pick the best model, meaning the model at the minimum, not when it was increasing again. So load best model at the end, meaning that you will pick the one with the best loss during all of the epoch that you've done. So FP16 means that you will use a precision of that takes 16 bits in memory instead of 64, for instance. So it's a way to save memory. And you lose an aggregate accumulation step of 50 or so. It's a way to save a lot of memory. Meaning that it's not at every batch that you will backpropagate your gradient, but you will wait for 50 batches to backpropagate your gradient. So you can play with all of these parameters if you want. Just wanted to say, the idea is on your side, you can change everything. So these are all of the parameters that you need for not the parameters of the model, of course, not the parameters of the LLM, the parameters of the training function of the trainer. Let's call it trainer. So here is the most important part. So here is the pre-training. So for that, what we'll use a function trainer, and it takes as input the model that we built previously without weights. I'm just saying it's a model that has an architecture, but the weights, there are no weights attached. This is training arguments. Okay, so this is what we just did before. The data collator function, this is a function that will feed the data to the model, the tokenized data to the sequences to the model. Here's the training set. Okay, so this is the data that will be used for computing the gradient, and the validation set will be the one that is used to validate at each epoch, to assess at each epoch the prediction accuracy. So you want to do that on a dataset that is independent from the one that was used for computing the gradient. And this function, stopping callback, early stopping patient equal 3, means that you know that your gradient is, I mean, we minimize the gradient at each epoch. We try to minimize the gradient, okay? And then at some point, the gradient will go up again. We want to take the model parameters when the gradient achieves a minimum, okay? So a local minimum. By using a patient of 3, we will try to um, prevent the effect of the noise during training, because it's kind of a bit noisy. When you minimize the gradient, it does like that. There is some noise, and it is possible that at a given step, you think you found the local, I mean, a good minimum, but actually, if you run 2 more epochs, you'll find another minimum that is better. So you don't want that this kind of noise that is inherently due to the data that you have, uh, to prevent you from finding a better local minimum. So use a patient. So for instance, by using a patient of 3, it means that although I found a good minimum, I can wait for 3 more epochs that will provide you less interesting loss, meaning a loss that is a little higher than your minimum. You wait for 3 more epochs until you decide that you stop, meaning that if after 3 more epochs, my model was not more, I didn't find a better model, meaning that I didn't find a model with a better loss, with a lower loss, then I will stop. But if during the 3rd epoch, I found a better model, meaning a model with a lower loss than the one that was the best, I will continue. So so what do we have? Oh, I have this problem, but I didn't have that last time. I'm just running the trainer now. Trainer.train means that it's training. So the idea here is not to train the model until the end, because it will take too much time, and actually Google will stop at some point you from using the GPU. So here is a trainer. You can see that there is 50 epochs, and we just did 2% of 1 epoch. And we'll estimate the time it takes per epoch, so you have an idea of how much it takes. So let's just run a little bit, and you can see that it takes time. We don't have yet an estimation of the time. Now we have an estimation of the time. So with this very small model, with this very small amount of data, it says that the estimation is for running 50 epochs is 20 hours. So keep in mind that it's not because I set the number of epochs at 50 that the model will be trained on 50 epochs. It's likely that it will stop before. So 50 is the maximum number of epochs. And what the model will do is it will stop, which is called stopping callback, basically. It will stop before the 50. So if it achieves the minimum before 50, so meaning that the function is doing that, then it's increasing again. The loss is doing that, and then it's increasing again. It will stop before the 50 epochs. Okay, so it's more like a maximum that you give, And here you can see that it's gonna take, so it means that it will take 20 hours maximum. Like it's likely that the model will require only half, maybe 25 epochs or 10 hours. Okay. So we're training. I'm gonna just do wait a little bit, and then we'll stop because we're not gonna wait for 10 hours for training the LLM. Let's wait a little bit. And then what we're gonna do, we'll cheat a bit by loading a model that I've trained already from my GitHub here. So we'll train the model Mistral DNA, so meaning that it's a mixed-run model that was pre-trained on the whole human genome. That's a model that contains 17 million parameters, and it was trained on the human genome 38 assembly. And if you want to see more information about this model, you can just look at my GitHub. Everything is free. You can use it as much as you want. And so, for instance, you can see my model here. There is a short description of the model. Actually, here it shows that this model was not pre-trained on DNA sequences of 200 bases, but it was pre-trained of sequences of 10,000 bases, so 10 KB. So the advantage of this model, it will process a larger DNA context, a larger DNA sequence. So it shows you how do you load this model here, how you can compute the embedding. Here I wrote a protein sequence, but it's a DNA sequence. I will have to modify that just right now. And that's it. So here it's pre-training. I wanted to look at the first epoch to show you the validation loss, but it's taking a bit of time. Yeah, one epoch. So you can compute that, for instance, 50 epochs takes 20 hours. It means that the time for an epoch is 20 divided by 50, meaning 20 minutes. So it takes time. So we'll not wait. And then I'm gonna just stop it. Okay. And then what we'll do is we'll cheat. We'll just assume that we pre-trained the model and we're gonna use it. But actually we're gonna use another pre-trained model. So let's load it. So this model, when we train it, it means that we download it from Hugging Face. And how do I do is I give this path here, Rafael Murad / trial DNA V1 17M AG38. So I give this path. I can copy it from here directly. I give it here. It will download the file from the Hugging Face server. So it downloaded the config.json, so the model architecture. Model.safe tensors are the model weights. And here you can see it has 8 transformer blocks, transformer layers. It encodes the DNA sequence with 4096 types of words, kinds of words. And let's compute the embedding. So what we can do with this kind of model is something that is kind of interesting is we can convert the DNA sequence to a vector. So let's do it. So I just computed, I took this sequence, I tokenized the DNA sequence, and then I computed the output of the model. Here's the output of the model. So the last hidden states, the last hidden neurons, last layer. So it will provide, first of all, if I look at hidden states, it will provide, let's look at the dimension. So the dimension 1 is because I have one DNA sequence, okay? And then 17 because This is due to the this is a DNA sequence that is converted to words that are larger than one base. So it corresponds to 17 words. Okay, and this is the dimension of the hidden state, hidden neurons. How many hidden neurons do we have? I mean, sorry, this is the number of words. Yeah, this is a dimension of the token space, meaning that we have 4096 possible tokens. So this is the dimension of the tokens. Okay. And then what I'm going to do is, for instance, I will use mean pooling. So meaning that I will take the average over the different position of the sequences. So here, the dim equal to 1, it corresponds to 17. So this is a vector here. If I look at embedding mean, this is a vector representing my DNA sequence. So it's a numerical vector, and it has a dimension of normally it has a dimension of 4096. Yes, because this is the number of tokens so this is the dimension of the tokens. Okay, so this is a way to convert a DNA sequence to this is a great way to a numerical vector. Okay, and when you use something like ChatGPT, actually what it does is you write a prompt, and this prompt is converted to a vector like that, exactly the same way, to a numerical vector. So I can use a mean pooling, I can use a max pooling. Let's look at the max pooling. It's a way to summarize information along the DNA sequence. And that's it. So, and then we'll see in other videos how to use this pre-trained model. So I hope that it was clear and very useful. This video was clear and very useful. Uh, it will be posted on YouTube. You can comment it so that I can improve it. And I thank you very much for your attention. Thank you. See you next time for the next video on fine-tuning DNA sequence for classification here.